Hello and welcome back to the first episode of Researcher Radio in 2019. Obviously we've been off for a bit celebrating Christmas and New Year, so I hope everybody had an amazing Christmas, a happy New Year, and I'm wishing everyone an extremely prosperous 2019. So for those that may not have listened to Researcher Radio at all in the 2018, this podcast is basically your regular look at research that's making waves in the scientific community and the authors of those papers. So my name is Joe Fenton, and as last year and into this year, I will be your host again. So today, I'm delighted to be joined by Dr. Sumitha N from the University of Chicago. So Sumitha is the author of The Molecular Genetic Basis of Herbivory Between Butterflies and Their Host Plants. Today, we'll be finding out a bit more about both the paper, the person behind it, and also how they deal with the pitfalls of academia and productivity in academia. So Sumitha, thank you so much for joining us today.
Thank you, Joe. Thank you for having me.
So, I've just introduced you, and obviously you're at the University of Chicago. But before we talk about your paper, could you give us a bit of a background on yourself? What's your academic journey been like so far?
Sure. I'm a broadly trained biologist who combines tools from various disciplines like molecular biology, microbiology, population genetics, and genomics to study the intricate genetic network between the plant host and its symbiont or pathogen or herbivore. So these are all like biotic and also abiotic interactions. I did my PhD in plant biological sciences at the University of Minnesota, Twin Cities. My PhD thesis was on the expression, regulatory, and evolutionary patterns of a large family of genes called fenson-like genes that are found in Medicago trincatula. Medicago is a model legume. Then I came to Joy Bergelson's lab at the University of Chicago for a postdoctoral project on resistant genes, or commonly called as R genes. These genes belong to the plant defense family in Arabidopsis thaliana. Again, this Arabidopsis is considered as the model plant among all the plants. So then next I moved to Cronford's lab in the same department and started working on different projects. Though the main project has been the molecular genetics of herbivory between the butterflies and their host plants.
And so what got you fascinated and interested in this topic overall in the first place? And that could either be, this particular project or what you study in general.
So I decided that I would work on plants when I first started thinking about my PhD thesis. So I was looking for opportunities in, the plant systems as models. And then when I started looking at like the different kind of projects that were available, I found this project where the plants, how they were interacting with their symbiotic partners, the rhizobia, and how these whole big family of genes that are, found in these plants are helping the plant to interact with these symbionts. So, and then once I was done with my PhD, I continued working with the interactions of plants with their pathogens. And right now I'm working with the plants, their interactions with their herbivores, which is like the insects. And now I have currently moved on to the plant and the abiotic factors, like, the environmental factors like CO2 and temperature. Just this whole concept of how the plants are interacting with their environment, though be it abiotic or biotic, was very interesting and appealing to me.
Okay, so you recently published this particular paper in Nature, Ecology and Evolution. And so for those that may not have read it or might not follow this particular journal, could you give us an overview of what you accomplished in this paper?
Sure. So the host plant and butterfly herbivore interactions have been the model system for the concept of coevolution. During the course of coevolution, plants are thought to continuously strengthen their defense repositories, and herbivores, in turn, follow suit with countermeasures to override them. So it's like a war arms race between the plants and their herbivores. So it's like a very coevolutionary arms race. Prior to our work, there were some interesting studies on the genetics of plant-insect interactions in the plants and also insects, but in separate studies, never together. Our paper is one of the first to study the molecular mechanisms in both the host plants and the herbivore insects simultaneously using genomic tools like genome-wide studies and transcriptomics in the model plant Arabidopsis thaliana and its herbivore butterfly, the Pyrus rapi. It's also commonly called as a cabbage white butterfly. Parallel genome-wide studies in Arabidopsis and cabbage white revealed a set of candidate genes for herbivory in both insect and plant. Analyzing the whole transcriptome over the time course of the feeding interaction showed that a large set of genes are being regulated in a dynamic way. And further comparative transcriptomic analysis, which included 4 diverse butterfly or plant systems, showed a variety of genome-wide responses to herbivory and also identified a core set of highly conserved genes in both butterflies as well as their host plants.
Okay, so I just want to pick up on the idea of the comparative transcriptomics within the paper and ask you if you can explain this at all.
Mm-hmm. Yeah. So when we did Arabidopsis and thaliana and Pyrus butterfly, time course transcriptomics, we found that there was a strong response of Arabidopsis to the butterfly eggs. So we wanted to see if this was a general pattern between the butterfly-plant interactions, or was it very specific to Arabidopsis and Pyrus? So we expanded our analysis of gene expression to 3 additional plant insect systems. Medicago sativa, commonly called as alfalfa. It's a huge forage crop in the U.S. Midwest, and its butterfly herbivore, which is Coleus urethine. This is like the clouded sulfur butterfly. The other system was Citrofortunella, or like the citrus family that has like oranges and limes. And their butterfly herbivore, which is the Papiliopolides, are commonly called as swallowtails. And Passiflora, the third system was Passiflora, also called as passion fruit, and its butterfly, the Heliconius signal. So, these were like a different, coming from different evolutionary clades, and were easily available also. So we compared the differential expression among all these different systems, and we found that there is a substantial variation in the responses of both plants and butterflies in all these systems. There was nothing similar, very, very different. And to further investigate if there were any components of the defense network that were conserved among the plants or among butterflies, we extracted and compared the common differentially expressed genes across the 4 systems. A small set of genes were found to be conserved among the 4 plants, and the majority of these genes, these conserved genes, had putative roles in defense systems. Similarly, even among the butterflies, there was a small set of conserved genes, among which Osiris-9 stood out because it was upregulated in all 4 butterfly species. Now, the function of Osiris-9 is unknown, but recent studies show that the Osiris gene family is involved in the detoxification of the plant host toxins in Drosophila. So these were very interesting. All these were very interesting results that we found by doing the comparative transcriptomics.
Okay, and I'm just this next question, I'm gonna go back into the paper, and, some of these questions that I ask our guests that come on may seem simplistic or not worth mentioning at all, but for me, I'm just curious to know why you used the offspring of wild-caught females for this particular study.
Yeah, that's a good question. So, apart from the transcriptomics, we also used the genome-wide analysis as a tool to find the candidate genes, right? So, we were investigating the genetic basis of the host plant-insect interaction, utilizing the natural variation that is found in the population in a GWAS, or the Genome-Wide Association Studies framework. GWAS is a powerful tool to characterize the genetic basis of phenotypic traits using the natural variation found in the populations. So in our experiments here, for the plant GWAS studies, we used 96 naturally available accessions of Arabidopsis thaliana. These were collections from different parts of Eurasia and North America, and a single lab strain of the Paris butterfly. And for the butterfly GWAS, we couldn't get butterflies from different parts of the world, but we just decided to go around like different parts of the Midwest in US and catch the wild females that were available to increase the natural variation in our study. So we caught 96 females, or females in specific, because we wanted the caterpillars or the larvae of these butterflies for our experiments. So the phenotypes that were used in our experiments were the amount of weight gained and the amount of leaf surface eaten by the 5-day-old larvae over a period of 72 hours. So that's why we collected females in specific.
Yeah, because in my preparation for this particular episode, I managed to find the video produced by the University of Chicago and the Confrost lab. And I saw, you guys running around trying to catch these butterflies in the hard pike area and in your campus. And it did look like a lot of fun.
Yeah, you could see that there was this nice video that was recorded when we were actually going around catching butterflies. Many people found it amusing for the scientists to run around catching butterflies, but it was fun.
Yeah, it really did look like a lot of fun.
Yeah.
Okay, so moving away from the practical elements, or the actual catching of the butterflies used for this study, I just wanted to talk a little bit about the use of next generation sequencing used in your study. So what is it and how did it affect this particular paper?
Mm-hmm. So this is a heavily genomics based paper. First, we sequenced and assembled a high quality Paris genome. And then for the GWAS, we resequenced 96 Paris larvae. For the transcriptome studies, we de novo assembled all the 8 transcriptomes of the 8 plants and butterflies that we were using in the comparative studies, and then we also sequenced the RNA-seq libraries, which were generated from 154 samples. So that was a lot of next generation sequencing that was used in the study.
Okay, so knowledge is a powerful thing, and everything in academia tries to influence something, whether that be in academia itself or in the real world. So I'm just curious to know, how do you think this piece could impact either the real world or academia itself?
Mm-hmm. So as I was saying, using this next generation sequencing, we generated this big wealth of information of the candidate genes and the putative genetic networks that are involved in the plant-insect interactions across the evolutionary clades of the plants and insects. So, the study has now opened doors to further functional characterization of the candidate genes that were found in the paper, and also will, can help in the fine tuning of the defense network systems of the genes that are found in plants and insects. And for the real world impact, I would say Arabidopsis thaliana is a small flowering plant that has been widely used as a model system. It belongs to the mustard family, which includes agronomic cultivars such as cabbage and radish. Pyrus rape is a common pest on the mustard plants. This butterfly is widespread across the temperate regions of North America and around the world. The larvae of these butterflies feed on the host plant foliage. They bore into the heads of these, like cabbage heads, and damage the most marketed parts of the plant. So there is like a huge loss, to all these crops. So the real world impact would be obtained by modulating the genes or genetic network that are implicated in the plant insect interactions to generate disease-resistant plants or crops and avoid huge losses in the agriculture to the pests.
Wow, that's really, really fascinating, because with my academic background, I understood the paper, but I couldn't make the particular link that you have just made, or I could not visualize it or conceptualize it in any manner whatsoever.
Mm-hmm. So, though Arabidopsis is like just, a scientific, used in the scientific labs, it's not like a crop or something, is considered as a weed. We also use like, agricultural crops like citrus, which have like economic value, Maricago, the alfalfa crop. And, this can be like extrapolated to other crops, species also. So,
Okay, and as I mentioned in the introduction to this piece, you published it in Nature, Ecology and Evolution. So I'm just curious to know how the publishing process was for you. Was it a smooth, or was it tricky? Like in many cases, quite a lot of academic publications actually are.
Yeah, I think it wasn't difficult to get the paper published. I would say it was pretty smooth. Though there were my other papers where it was kind of a rough ride, but for this paper, I think we were fortunate that it was pretty smooth. We first submitted the paper to Nature Genetics, who very politely asked us to consider submitting the paper to Nature Ecology and Evolution because the editorial board thought it would be a better fit there. And in Nature Ecology and Evolution, we had some very helpful comments and suggestions from the reviewers that helped improve the results and discussion sections of the manuscript.
Okay, so overall not too bad. Because obviously, speaking to some academics, they've told me some absolute horror stories from some of the papers that they've submitted to particular journals.
No, it wasn't bad. I would say it was smooth. But then again, there were other papers in my career where it was a little bit rough and tough.
Okay, so submitting a paper obviously takes time to make and build, and obviously you have to create experiments. You've got to keep up to date with the literature. You've got 1,000,001 other things to do. So I'm just curious to know how you usually spend your week.
Mm-hmm. I believe in a good work-life balance, but there are situations when work takes over the family time, especially like when you get stuck on a step in data analysis, or you're trying to troubleshoot a protocol in your experiments. Also, paper and grant writing take up a lot of family time. So, I try to maintain the balance, but sometimes the balance gets offset by, different situations at work.
Okay, now obviously, so the metaphor goes that there's a sea of literature out there, and I'm just curious to know, how do you keep afloat in it, or is it a struggle?
Mm-hmm. Mm-hmm. Yeah, I think spending time on latest literature is very important. Um, I have online subscription to some top journals in my field. So I get weekly or monthly emails about the latest papers. And then social media platforms like Facebook and Twitter are also helpful. You can actually follow different scientific groups on these platforms to keep you updated on like new papers and new research. And I also think like academic podcasts like, Researcher Radio are also a good medium when you don't have time to sit and read, but you can just listen to podcasts while driving or like doing lab work. So, on average, I think I spend around 4 to 6 hours per week reading or listening to various media to keep myself updated on the current scientific trends.
Okay, so a lot of this particular podcast seeks to aid those that may have faced problems with productivity. And so I'd like to ask all the academics that come on as guests here if they've got any tips that they've used to increase their own productivity or academic output.
So I think I consider myself very organized. So, and I think it has been a major strength for me, especially when I have to balance multiple things at the same time. So I would say being organized, planning your schedule, prioritizing the needs, these are all correlated and will help increase the productivity.
Okay, and so your field in general, if we go back to the actual raw science, what for you is up and coming, or do you find really interesting that's occurring in your field right now?
So more than topics, I would like to talk about this tool, especially in biological sciences, the CRISPR. The CRISPR tool and the gene editing tools are revolutionizing a lot of fields among biology and medicine. In medicine, there are trials on genome surgery that are being carried out in different labs right now. The whole genome surgery is, the precision targeting of genetic defects in the genome using the CRISPR-Cas9 and can be customized to individual patients. And this CRISPR can be, even in agriculture, can be used to generate more successful and stable GMO crops. So I think it will be interesting to see how the different parts of the world will come up with regulations on these CRISPR modified products.
Okay, and so what about yourself? What projects or papers have you got in the pipeline?
So I'm working. I'm continuing to work on the different projects, but my current work right now is actually the continuation of the work that was done in this paper. We are studying the effect of the different environmental factors like CO2 and temperature on the genetics of plant-insect interactions. So what would happen to the whole genetic network if we change, say, the CO2 levels or the temperature levels, or both CO2 and temperature together? So we have done those experiments, and right now, currently, I'm analyzing the data and hope to get the paper out pretty soon.
Wow, that's fascinating, because I'm extremely interested in, as you said, ideas of warmth and CO2 and how that affects the planet, especially as we go into an age, or we're now in an age of, environmental enlightenment, or there's a consciousness around protecting and saving the planet.
Yes. Yeah. Mm-hmm. Yeah. No, I mean, very current about like the whole climate change process, things that are happening. So how that will affect the plant insect interactions, like what will happen to the plants, what will happen to the insects, and what will happen to the whole interaction actually.
Okay, and as we're coming to a close on this particular episode, I'm going to ask the same question that I ask every guest that comes on, and that is there one piece of advice for anybody that's about to undertake a PhD or begin their career in academia?
I would would suggest someone who is thinking of doing a PhD to make sure the PhD program you're choosing has a variety of research options, and to start interacting with as many research groups or teams in your field or department as soon as possible, right from the start, I would say, because research is moving towards more interdisciplinary projects. Effective collaborations and network have become a key for a successful academic career. So, start networking, talking to different scientists, fellow or different mentors, and just network.
Yeah, and I find this point extremely fascinating, and one that I've never really thought of before. But speaking to the academics that come on this podcast, they've all said that, the world of science is opening, and it's opening quicker than it ever has done before. And there's so many cross collaborations across different subjects and departments.
Yeah. And not one single person can't do everything, right? So, of course, you are expected to do quite a few things, but, you also have to keep learning and, collaborate with different people to do this big interdisciplinary project. So.
Okay, so that's all we've got time for today on Researcher Radio. So we've been joined by Dr. Sumitha N from the University of Chicago. Sumitha, thank you so much for coming on and speaking with us today.
Thank you for having me, and it was a pleasure talking to you.
And thank you for listening, everyone. Until next time. Okay, and before I leave you guys, I just wanted to thank you so much for your reviews and your suggestions for the podcast. And moving on and moving forward, I will take your suggestions into account. So again, thank you for sticking with us, and I'll see you next week. You've been listening to the Researcher Podcast. You can follow us on Twitter, Instagram, Facebook, or LinkedIn. You can also follow us online at www.researcher-app.com. Or, alternatively, you can drop me an email at joseph.fenton@researcherapp.com. Researcher is free to use on iOS, Android, or on your web browser. And if you enjoyed this podcast, don't forget to leave us a review.